Hello everyone. Welcome to ITLS Academy Empower the Youth. Today in this lecture we will going to study about microbiology. What is microbiology? Why microbiology is so useful and why there is a need to study microbiology and how means from where this microbiology came into existence. So, students, we all know that this microbiology is nothing but it is a study of tiny creature, or you can say small organisms that you cannot see with your naked eyes. You need certain microscope to visualize that organism. So, this study about these organisms are known. This study, which is dealing with this microorganism, is known as microbiology. So, in this, we will going to study about the detail of microbiology. So, quickly, let's see what all we will going to study in this lecture. The first is what is microbiology? Second is why it is such an important subject and how have we gained our present knowledge of microbiology. So, as already told you that this microbiology it is nothing but it is a study of organisms. Why it is such an important subject? Because in life sciences courses, in biotech courses, in any btech courses, even in the different bsc, msc, or even in the pharma also, this microbiology subject plays an important role. So what are the importance of the subjects? We will going to study that, and we will see what we have gained, our how can we gain our present knowledge of microbiology. So let's get started. So what is microbiology? So microbiology should be an easy word to define: the science logos of small micro life bios, the study of living things so small that they cannot be seen with the naked eyes. So what is microbiology? In simple word, if we say, it is a study of, it is the study of living things, means organisms so small means that organism that you cannot see with your naked eyes. You need a microscope to see these organisms. So I'm taking this name organism. So organism is a living creature. In biology language we say this organism as a microorganism. Why microorganism? Because they are very small, right? So this study is known as microbiology. Okay, so bacteria neatly fit in this definition, but what about fungi and algae? So students, if we talk about bacteria, we all know that this bacteria are very small creature, isn't it? That they cannot be seen with your naked eyes. If I am sitting here, so there are number and numbers of microorganisms, or you can say there are numbers and numbers of bacteria that are present on my skin, that are present on my laptop, that are present on my desk, right? But I'm unable to visualize this bacteria, right? But if I take a small sample of my skin, or you can say any sample of a table, and if I see under a microscope, then I can simply say, yeah, there are thousands and thousands of bacteria present in a very small area. But if we say about fungi and algae, because you can see fungi and algae with your naked eyes, then how this definition fit for fungi and algae? You have to answer this. Ki why fungi and algae, because fungi and algae you can see with your naked eyes, right? But why it is known as microorganism? Because microorganism means uh, a, a creature that you cannot see with your naked eyes, right? So why this fungi and algae are important, sorry, plays an why they are known as microorganisms, right? So to the answer to this, I'm writing on a blank page, that is blank screen, sorry. If we talk about this bacteria, so they are small, but if I am talking about this fungi and this fungi and algae, so this fungi and algae, why they are considered as microorganism? So simply, if we talk about this fungi, so if we talk about the fungus that grows on bread, so they cannot be seen with your naked eyes. If you see the fungus, so if you want to see the fungus, right, so you need a magnifying glass to see this fungus, right? So that is why it is considered as microorganism. Similar case is with this algae also, right? So algae have a nucleus, okay, that exists as a microscopic cell, and they generate oxygen through a photosynthesis. So here the nucleus of the algae is exists as microscopic, right? Now, coming to the next, that is, these two groups each contain members that are far from microscopic. On the other hand, certain animals such as nematodes, worms can be microscopic yet are not considered to be a domain of a microbiologist. Viruses represent another special cases. They are most certainly microscopic, but by most accepted definition they are not living. Okay, so apart from this fungi and algae, there are certain other animals such as nematodes, worms, or you can say there are certain viruses, right? They are also considered as a microscopic. But if we see the domain of the microbiologist, they do not consider this worm under the heading of microorganisms. So there is a quick changes. Now coming to why microbiology is important, why it is important, why there is a need in every courses, in every stream microbiology subject is there, why there is an importance. So as we have discussed, this microbiology means the study of sinister, or you can say when invisible bugs that cause diseases, means microorganisms, right? So microbiology played an important role in the formation of medicines, environmental sciences, food and drinks product, fundamental research, agriculture, pharmaceutical industries, genetic engineering. A quick example if I give you of curd. Curd is made by the fermentation of, that is done with the help of microorganisms, right? So this microorganism plays an important role in every, you can say, every field, right? So you can also say that if I talk about cheese is also be prepared through microorganisms, right? So microorganisms are playing a very important role in every, you know a food or a drink production. Whereas if I talk about this medicine and pharmaceutical industries, so how they are useful, how this field is important. If I talk about this pharmaceutical industries, so if we say, by understanding the principle of microbiology and human cell mechanism, so they allows pharmacists to discover antimicrobial drug that would prevent an accelerating number of communicable diseases. So they are allowing the pharmacist to discover what antimicrobial drug that would prevent the future कह सकते हैं कि accelerating number of communicable diseases. So in this whole series of microorganism, I am going to be using this term, that is pathogen, in every or a second line. So what is pathogen? So pathogen is nothing but it is an organism with the potential to cause diseases. So pathogens are those organisms they that causes diseases. Now moving to the next, that is, how do we know microbiology is perceptive to the golden age and beyond? So microorganism had been on earth for some 4000 million years when Anthony von Leeuwenhoek started out on his pioneering microscope work in 1673. Leeuwenhoek was an amateur scientist who spent much of his spare time grinding glass lenses to produce a simple microscope. We all know that the discovery of microscope is done by Antony von Leeuwenhoek. So he started his microscope work in 1673. This questions generally arises in MCQs when he started his work. So 1673 में he started his work on microscope for microscope, and this microorganisms had been on earth for about 4000 million year ago. But before the discovery of microorganism, nobody was aware of what are microorganisms, right? So this Leeuwenhoek was an amateur scientist who spent much of his spare time grinding glass lenses to produce a simple microscope. So he simply, when he was free, in his free time, he generally, what he do, he does the grinding glasses and lenses, and with the help of this grinding lenses, glass lenses, he produces a simple microscope. Nowadays there are number and different types of microorganisms are present in the on earth. So if in Pasteur world, if I say, the doctrine of the spontaneous generation will never recover from this mortal blow. There is no known circumstances in which it can be formulated that microscopic beings came into the world without germs, without parents similar to themselves. Pasteur findings on wine contamination led inevitable to the idea that microorganisms may also be responsible for diseases in humans, animals, and plants. So, this Louis Pasteur was another scientist who stated that these microorganisms also see, before 4,000 million year ago, microorganisms were or present on the earth. So it is after the discovery of this microscope, that is done by Antony von Leeuwenhoek, the study of microorganism came into existence. But microorganism may cause diseases, that was told by Pasteur, right? So he stated that microorganisms may also be responsible for diseases in human, animals, and plant. So if we talk about the different types of microscope that I was telling you, that is, microscopes are of different types, right? If if we see the classification of microscope, so microscope is divided into different part, that is light microscope, electron microscope, right? So light microscope is further divided into two subcategories, that is compound microscope and stereo microscope. If I talk about the compounds microscope, it has a fine slice, or you can say a section of specimen required. Light passes through a specimen, image appears 2D, stains are often required to see the details. Then if I talk about the stereo microscope, then whole or a part of a specimen can be viewed. Light bounces off the surface of specimen, image appears 3D, viewed with natural colors. If I talk about the electron microscope, then scanning electron microscope means whole or part of a specimen can be viewed. Electrons bounce off surface and are detected. Image appears 3D, image in grayscale but can be colored. This is about this scanning micro electron microscope. Then TEM, that is STEM, that is transmission electron microscope, a very fine section of a specimen required. Electron passes through and interact with the specimen. Image appears 2D, image in grayscale but can be colored later. So different on this slide, if I talk about this slide, so basically there are some questions that generally arises in exams. They can also ask you about what is the difference between a compound microscope and a stereo microscope. In exams they may also ask about scanning microscope and transmission electron microscope. This question 90% there is a chances of this question, what is the difference between, I'm writing some questions at the end of this lecture. You can simply go to that. What is the difference between SEM, that is scanning electron microscope, or TEM, that is transmission electron microscope. Now moving to the next, that is a light microscopy. That is a light microscope. So this is how the light microscope looks like. It has an eyepiece from where you just generally see. Then it is an arm. This is a nose piece. This is an objective lenses. It is a stage where you place your slide. This is a diaphragm for the adjusting. This is a coarse adjustment, sorry, and this is a fine adjustment. This is the source of light, that is light source, and this is a base. So light microscopy, it is a small specimen are made visible by throwing light into a teen beam and then creating an image to understand how the structure interact with the light, right? So in this microscope, basically a beam of light, you can say throwing a light into a team beam, and this image, how the image is created, this image is created, how created to understand, means this image which is being created, so it is created to understand that how the structure interact with the light that is falling on it. Some parameters like reflection, absorption, and scattering of the sample get discovered. Now, this is a light microscopy. Now coming to the electron microscopy. So electron microscopy, that is known as EM, is a technique for obtaining a high resolution image of a biological and non-biological specimen. So basically in this it has a as compared to the light microscope, it has a high resolution image of both biological or you can say a non-biological specimen. It is used in a biomedical research to investigate the detailed structure of tissues, cells, organelles, micromolecular complexes, The high resolution of images result from the use of electron, that is, which have very short wavelength as the source of illuminating the radiation. So basically it has a more advanced feature as compared to the light microscopy. This microscope is used in different biomedical research, where you now want to get the detailed structure of any tissues or cells, or you can say organelle or molecule macromolecular complexes. So this is how this electrons basically work. In the next lecture we will going to study about the detail how the you where you put the slide, how it generally works, right? How the image is created. So this is all about the overview of microscopy. So this is all about today's lecture. At the end of the lecture, what I'm planning, I'm giving you some questions. These questions may be asked in exams. So simply, there are different types of questions, right? Where you can, simply go through this and get some answers. So I'm writing some. So the first question generally arises that what is microbiology? The first question is what is microbiology? Right? This question generally arises in exams. Second question is why, or you can say not why, they can ask you. Second question I'm writing here. Oh, sorry. Again, I'm writing. First is what is microbiology? Second is application of microbiology. Lastly, difference between SEM, that is scan emission microscope, and yeah. So the last is difference between SCM, that is scanning electron microscope, and TEM, that is transmission electron microscope. Sometimes they also ask you the difference between light microscope and compound electron microscope. So these are some important questions on the basis of the slides. You can simply take a screenshot, or you can simply note it down somewhere. So because this in this these are some important questions that generally asked in exams. So students, hope you have understood the lecture. If any problem related to any slide, or related you want me to repeat that slide, you can simply mm, uh, drop your questions in the comment section below. If I be receiving any questions, I'll be discussing in my next lecture. So stay tuned and don't forget to subscribe ITLS Academy. You can also follow ITLS Academy on Twitter, Instagram, Facebook, YouTube, and please subscribe ITLS Academy. Press the bell icon, and you can also visit our website, that is www.itlsacademy.com. This is our helpline number, that is 7080 7080833450. If you have any queries related to lectures, related to any course, you can simply call on this number and get updated. Thank you for watching. Please, student, do like, share, and subscribe. Thank you.